everyone. I'm the lucky last talk today, and I'm from BABS, which stands for Biotechnology and Biomolecular Science. In the loose sense, bees is kind of bigger animals like giraffes or even plants, and in BABS we deal with microorganisms down to the protein scale. So I'm actually a biophysicist, which segues in from the quantum talk we just had, but I'm a little bit more on the biological end of things, and I particularly study force in biology. On the one hand, that's sensing force, which of course is of interest when we hear this talk or when we touch things, or also in other organisms in pressure response. At the other end of the scale, we have using the force, like Yoda here, which of course is informed in my muscles. I have bundles of molecular motors that contract to allow me to wave my arms and talk. And we also have molecular motors, which are actually things that convert chemical energy into a mechanical force. So the one that I spent most of my career studying is this bacterial flagella motor. It's a tiny rotary nanomachine that can build itself in a bacterial membrane, rotate 5 times faster than a Formula One engine, change direction in thousandths of a second, and navigate bacteria on average to where their life is better. But we effectively studied this particularly from the assembly angle. Like, how is it that it self-assembles? That's quite foreign in that if we throw a bag of Lego on the ground, we know it doesn't spontaneously form a pirate ship. But in the case of the bacterial flagella motor, that is exactly what happens. It spontaneously forms in the right time and place. So that was some structural work looking at specific structures of those components to work out how that assembly and how that force generation occurred. But what I'm going to talk about now is the other end of the scale, sensing force. So I talked about touch and hearing in higher organisms, but actually in bacteria it's pretty important too. Because, for example, when it rains, there's a huge change in the external concentration of ions in the soil. So if bacteria didn't have some pressure response or pressure release valve, they'd actually just explode when it rains. So in bacteria, this is the MSCS for small and MSCL for large channels that effectively control that pressure response. And in communities, they're also important for biofilm formation. But in big, complicated things like us, they're obviously involved in many things, in cell-to-cell junctions, cell surface interactions, and a lot of physiology, such as cell division, actually physically pulling a cell apart. Now that's the physiology end, but obviously there's the pathophysiology end as well. So what we call mechanobiology is actually very important. Cancer cells are typically much softer than normal cells. Cancer detects gaps in order to invaginate, and also things like differentiation of stem cells. Engler showed in 2006 that if you just take a stem cell and change the stiffness of its external environment, you can actually differentiate it to become a stiff bone cell or to become a soft neuron cell. So clearly the cells can interact and sense how stiff their environments are, and it's quite an important pathway. So the whole field of mechanobiology, what do I actually look at? I look at this protein called piezo. So piezo is a new player. It's a bit novel because it was only discovered in 2012, and it was only discovered in 2012 because it's actually quite enormous. So we care about this on the pathophysiological end because single point mutations cause these blood disorders that are quite rare but are very specifically linked to piezo. But in the bigger picture, these are overexpressed in cancer and particularly involved in post-traumatic pain and osteoarthritis. Now this structure here is from something called cryo-EM you may have heard of, and comes from my collaborator at Tsinghua, Bailong Zhao. So it's worth mentioning, seeing as this is talking about scalability, one of the aspects of scale is going to smaller and smaller scales. So cryo-electron microscopy, you may know, was actually the Nobel Prize in 2017 in chemistry. What that slide shows is that in 2013, this was mostly just blobs. It was called blobology, because effectively everything you looked at looked like a blob. So there was a lot of cynicism, but now in the last 4 years, we have really, really high resolution. So you can actually see almost the individual atoms involved. So it's really seeing is believing. And of course, Dubuchet, Frank, and Henderson got the Nobel Prize for freezing the computational and for doing it respectively. And this is technology that's really coming to BABS. So Niang Yan is one of China's most eminent young scientists. She's a specialist in cryo-EM. And this last year, she sold the structure for the first eukaryotic sodium channel, and then also did it in an electric eel. And you can see what I mean here by seeing is believing. You don't actually have to have much of a structural biology model to fit behind this because you can see the individual alpha helices of protein. So it almost does it itself. Of course, a lot of complex analysis and experimentation goes into getting this level of structure, but this is the type of thing that we're looking to take things to the smaller and smaller scale to understand exactly how they work. Now, what do I do? This is a lot of other people's stuff, but what I actually do is work on a technique called droplet hydrogel bilayers. So if we take a droplet and we basically touch it together to a lipid surface, we can make this artificial cell membrane on a transparent surface. Why do we do it on a transparent surface? Well, we do that primarily so we can interrogate it with a microscope at the same time as we measure the electrical signal across that bilayer. So why we care about that in a mechanosensitive context, or these force-sensing proteins, is that we can then inject more fluid into that droplet and we can measure the electrical signal. So this class of ion channel, a mechanosensitive ion channel, is one that when it senses a force, when you poke the droplet, it will open to allow current to flow. So there's an electrical profile behind that, and if you look at it in this system, you can actually also see the protein and see how its stoichiometry and its conformation changes. So again, talking about scalability, well, that's one droplet, one bilayer. Maybe you have a few proteins in it, but really where this wants to go is to multiple droplets, to effectively use fluidics to assemble multiple droplets, to have multiple bilayers, and start getting closer and closer to that larger scale sort of tissue, bridging that divide from a single droplet to the biology of tissues. So again, this is work from collaborators where you've actually almost 3D printed a droplet cube of about 1,000 or so droplets, and you can selectively insert membrane proteins into each of them in order to build a conducting channel through them. So we talked a bit about some industrial collaboration, and so in this case, this is work out of Hagen-Bailey's lab, which was alpha-hemolysin in these droplets, which was the origin of Oxford nanopores. So it's kind of making droplets and membrane proteins and functionalizing them to do, say, pore-based sequencing. So at the other end of the scale, on the high throughput thing, evolution is naturally a very good high throughput computer that's been running for billions of years. So what you're looking at here is actually work from Michael Bame's lab, where they made a meter-long Petri dish, and they made it 10 times stronger in antibiotics every 10 centimeters or so. Inoculating the plate at either side, you can see that actually it's almost very synchronized in the way that that antibiotic resistance evolved. So that's surprising. You probably would have thought that one of them overtook the whole plate more quickly than the other. But by then tracking that and looking back and analyzing how that evolution has happened, you can look at what constrains that space of evolutionary possibilities. So the link to the previous work on fluidics and the 3D gel printing is if you want to tinker with evolution and do it in a manual way, you need to have liquid handling and robotics, and you need to have sort of larger and larger scale of the automation that you use to apply that problem. So that's what we're bringing online to BABS as well. We're bringing a new course in synthetic biology in second semester next year. And of course, the third element of scale is just having new spaces to put this in. So this is the new E26 Biosciences building, D26, cleverly named. Next to it will be retrofitted, and that's where a lot of the structural biology and the synthetic biology capacity is going to go. So please check the website. Thank you for your attention, and thank you to all my other speakers for entertaining me.